everyone, my name is Neera Patadia and I'm a graduate student in the Pavlidis lab located at the University of British Columbia, and I'm going to talk a little bit about my project, Exploration of Large-Scale Differential Expression Datasets. In genomics, we're often interested in examining how genes are regulated among biological conditions. This can be done by performing a differential expression analysis, which involves comparing the expression of genes from some baseline condition to that of a condition of interest. Understanding this landscape of differential expression across multiple biological conditions can provide some valuable insights into understanding how genes are regulated among these many biological conditions. In this work, we leverage the GEMMA database developed in the Pavlidis lab. The GEMMA database contains thousands of high-quality human differential expression datasets, which have been manually curated using formal ontologies. We are interested in gaining a global overview on the types of differential expression studies that are in GEMMA and examining how they are related to each other in terms of their semantic contexts. Studies in the GEMMA database are scraped from GEO. This data that is scraped from GEO can be broken down into two key categories for each experiment, which is the expression data and the actual metadata. The expression data is reprocessed according to the platform used to generate the data, and outlier removal and batch correction is performed. When it comes to the dataset metadata, a team of trained undergraduate students curate the studies to best capture the experimental design of the study using formal ontology terms. The experimental factors represent the feature that is being varied across samples. So for example, a study examining the effects of a drug on different cell types would have the experimental factors treatment and cell type. These features would have levels or factor values that represent the types of drugs and the types of cells that are being looked at in the experiment. In order to perform differential expression analyses on these data, experiments are annotated with the baseline factor. When performing the differential expression analysis itself, we use lemma for data obtained from microarray studies and lemma-voom for RNA-seq studies. Currently, there are approximately 20,000 human differential expressions experimental contrasts in the database, with more data being routinely added. The majority of these datasets have been annotated using formal ontology terms. GEMMA uses several formal ontologies to manually annotate the experimental contrasts. When looking at how frequently a given ontology term is used to annotate the experimental contrasts, most are applied to only a single experimental contrast in the database. These annotations cover a broad range of topics, and we can see that studies examining disease conditions are highly represented. This means that GEMMA contains many experimental contrasts that involve looking at the expression of genes for some disease condition in comparison to another disease condition, or to that of some non-disease control. The topics that are examined among the experimental contrasts in GEMMA are incredibly diverse, as we can see here where the terms are scaled according to their frequency in the database. GEMMA contains many studies involving brain-related diseases and disorders, such as Alzheimer's disease, autism spectrum disorder, schizophrenia, and bipolar disorder. Cancer-related studies are also highly represented as well. Genetic manipulation studies, where the effect of a gene being say, knocked down or overexpressed in the experiment, are highly represented. These studies make up approximately 1,212 experimental contrasts and collectively look at the impact of perturbing 1,141 genes. Many of the manipulated genes featured in the database are transcription factors, such as RUNX1, HIF1A, STAT3, as well as known cancer driver genes such as TP53 and TP63. To examine how experimental contrasts are related to each other based on the semantic similarity of their annotations, we utilize the normalized term overlap metric from Mystery et al. To calculate NTO between two terms, the propagation path of each term is retrieved by visiting all associated parent terms, excluding the root term of the ontology. These sets of propagated terms are the annotation sets. The size of the intersection between these annotation sets is calculated and is further normalized by the size of the smaller of the annotation sets between the compared terms. Using ontology inference enables us to group experimental contrasts based on their semantic similarity rather than exact matches, which can guide downstream analyses. For example, in the left figure, we can see the more propagated terms that are shared between two annotations, the more semantically similar they are to each other. For the experimental contrasts in GEMMA, we look at the differential expression of genes by examining the genes that have an FDR less than 0.05 and a log base 2 fold change cutoff greater than 1.5 or less than negative 1.5. Different experimental contrasts will have different number of differentially expressed genes, which we can examine by looking at the distribution of the number of DE genes according to the original platform that was used to run the experiment. Here we can see that experiments conducted using RNA-seq have more genes that are differentially expressed in comparison to that of microarray studies. Furthermore, when looking at the distributions when broken down by the topic the experimental contrast annotation was derived from, chemical entity studies, that is, studies examining the effect of various chemical treatments, appear to have a fewer number of DE genes in comparison to the other major topics in GEMMA. Next, we look at how frequently genes are differentially expressed among all experimental contrasts. Furthermore, we also examine how the frequency of these genes map to the DE prior, a metric developed by Crow et al., which examines the predictability of human gene expression. Our data recapitulates the findings from Crow et al. As genes that are frequently differentially expressed across the contrasts have a more prominent DE prior. So to wrap things up, the GEMMA database contains rich data on the differential expression of genes in a diverse range of conditions. Going forward, we will conduct more in-depth analyses on the structure of the data in GEMMA. We hypothesize that this data could be used to identify recurring patterns of differential expression across biological conditions, which in turn could be associated with regulators. Finally, we will conduct parallel analyses using the mouse datasets within GEMMA. I would like to acknowledge our funding sources and support from fellow lab members that helped make this project possible. Thank you for listening.